In this video, we'd like to explain how to calculate ontologically grouped differential analysis. In the case of the bin discovered database, the data come from the GC bin-based database, which contains GC TOF metabolomics analysis for over 156,000 samples, 55 million annotations, and 2,000 studies. And for our purposes, each sample has three types of metadata associated with it: a species value, an organ value, and a disease value. And so essentially what we can think of the bin discovered database as containing is, for each triplet of metadata combination, for each compound, a distribution of annotation intensities. We can recognize that the species and the organs can be mapped to or linked to external hierarchies. For example, the species can be linked to the NCBI taxonomy, and the organs and diseases can be linked to the mesh hierarchy. Shown here is an example of a subset of the organ hierarchy. And so when we go to do ontologically grouped differential analysis, instead of simply comparing the distributions between two triplets or single metadata combinations, e.g. apple fruit without disease versus fig fruit without disease, we can instead compare categories like apple fruit without disease to all other species of fruit without disease in order to try to understand high confidence trends that make certain metadata combinations comparatively unique. How do we do these calculations? We'll explain this for one compound, although you would do this type of calculation for each compound that you're comparing. And so for the example, we will compare the human digestive tract without disease to bacterial cells without disease. And so we recognize that there are certain things in this query like digestive tract or bacteria that are categories, not actually metadata values for which we have data. And so we can turn to our hierarchies, for example, the mesh organ hierarchy, and recognize that at the top of this mesh organ hierarchy, we have this category that we're interested in, the digestive tract. And then in the leaf nodes, we have actual organs for which we might have data. And so we can take this hierarchy and kind of substitute it in for the category of digestive tract and rewrite the left-hand side of this query as a set of metadata combinations for which we do have actual distributions for this one compound. Likewise, on the right-hand side for the species, we can use the NCBI taxonomy, shown here in greatly simplified form, to replace this category of all bacteria with these different types of bacteria. And so we can rewrite the right-hand side of this query as as a set of metadata combinations for which we have data. Then, because we have distributions for each of these triplets for this one compound, we can do differential analysis between each element on the left-hand side and each element on the right-hand side. So we're doing pairwise differential analysis, and which means we're going to get a full change of averages between each element on the left-hand side, as well as a p-value between each element on the left-hand side and each element on the right-hand side. For the purposes of explanation, we can rewrite this calculation as two matrices, a matrix of fold changes and a matrix of p-values, where each element on the left-hand side is now a row label, and each element on the right-hand side is now a column label. And each of our pairwise calculations becomes a cell or a value in this matrix. Then, for the purposes of making a conservative estimation of this behavior, because we're interested in extracting small amounts of tractable data rather than keeping everything, we can take the minimum fold change value and the maximum p-value and use those as coordinates in the hierarchically grouped volcano plot. And if we have fold changes that are both positive and negative, then we can represent the minimum as 0. And so if we go to do this calculation between the human digestive tract and all bacteria cells, we see a handful of compounds that are increased in the human digestive system compared to all other bacteria, such as different types of cholesterols.